Every city or village produces millions of tons of wastewater, spent water from residences that carry body wastes, excreta, food remains, and laundry wastes, to name a few. Similarly, industries spew commercial wastes through wastewater. All such wastewater is called sewage. Have you ever wondered where all this sewage produced by us goes? It certainly cannot be just deposited as it is into rivers or the sea. Sewage is first treated by microbes that are naturally present inside it before it can be safely disposed of in rivers and streams to avoid polluting these water bodies. That is why the Ministry of Environment and Forests has produced the construction of a large number of sewage treatment plants under the Ganga Action Plan and Yamuna Action Plan to save major rivers like the Ganga and Yamuna. In India, microbes are already treating gallons of wastewater inside several sewage plants around the country every day. Let's take a look at how microbes help in sewage treatment. Microbial treatment of sewage usually takes place in two stages: primary and secondary treatment. In the primary stage, the objective is to remove coarse solids and other large and small particles found in sewage. The first step of primary treatment involves filtration, which is followed by grit removal inside a primary settling tank. All the floating debris is removed by sequential filtration, whereas the grit, such as coarse soil or pebbles, is removed via sedimentation. During sedimentation, most organic and inorganic solids settle, while floating materials like grease and plastic rise to the surface and are skimmed off. The settled solids are called primary sludge, while the floating material comprises primary effluents. Primary effluents are now transferred from the primary settling tank to large aeration tanks for secondary treatment, also known as biological treatment. Secondary treatment aims to reduce organic matter in the effluents. In the aeration tanks, the effluents are constantly agitated mechanically and oxygen is pumped into them. As a result, aerobic bacteria naturally present in the effluents grow into flocks, which are mesh-like structures formed by groups of bacteria in association with fungal hyphae or filaments. In the presence of oxygen, these growing aerobic microbes consume the organic matter present in the effluents. This secondary treatment reduces the BOD, or biochemical oxygen demand of water. BOD is an important measure of water quality. It refers to the amount of oxygen needed by bacteria and other microbes to oxidize all the organic matter in 1 liter of water. The BOD of drinking water should be less than 0.5. However, the BOD of raw sewage can be as high as 600 milligrams per liter. That is, the greater the BOD of wastewater, the more polluted it is. Also, it is an indicator of the amount of organic matter present in water. Secondary treatment is continued till BOD decreases considerably. Once this is accomplished, the effluents are transferred to another aeration tank where the flocks of bacteria are gradually allowed to sediment. This sedimented part of sewage is known as activated sludge, which is pumped into large tanks called anaerobic sludge digesters. Here, anaerobic bacteria digest the bacteria and fungi present in the activated sludge and produce a mixture of gases such as methane, hydrogen sulfide, and carbon dioxide. This mixture of gases is also called biogas and is used as fuel since it is inflammable. Methane is the main gas produced from sewage treatment and also serves as fuel. It is used to produce heat in homes and factories. The sludge left after digestion of both aerobic and anaerobic bacteria is used as manure and is called activated sludge. The effluents that now remain from the secondary treatment plant are now fit to be released into natural water bodies like rivers and streams. However, a small part of the activated sludge is pumped back into the aeration tank to serve as inoculums for subsequent treatments. Though the amount of sewage produced increases every year with the population increase and rapid urbanization, microbial treatment of sewage remains the most effective method to date. For thousands of years, since the time of ancient Egypt, yeast has been used for baking and fermentation. With time, it was observed that dough rose during the bread-making process due to the exhalation of carbon dioxide by yeast. When bread is baked, the carbon dioxide escapes, leaving a porous and light bread. Similarly, during fermentation, to prepare beverages and cheese, the main gas that yeasts produce is carbon dioxide. It is therefore evident that microbes produce gases during their growth and metabolism. Even so, different microbes produce different gases depending on the organic substrates they utilize. For example, anaerobic bacteria that grow on organic matter such as cellulose produce copious amounts of methane along with carbon dioxide and hydrogen gases. Due to this reason, these bacteria are collectively known as methanogens. Did you know that explorers have found methanogens even in extreme climates such as under the ice in Greenland and in the hot, dry desert of Utah in the United States? Cattle also have methanogenic bacteria called methanobacterium present inside the rumen. This helps them digest the cellulose present in the grass they feed on. This explains why cattle dung, also locally called gobar, is rich methanobacterium. This dung or gobar is used to generate biogas, also known as gobar gas, in a biogas plant. In India, two types of biogas plants are used, the fixed dome type and the floating gas holder type. In the fixed dome type, the dome is made of concrete and is fixed, whereas in the floating gas holder type, the lid is made of stainless steel and is movable. The floating gas holder type of biogas plant is covered with a floating lid so that it can freely rise when microbial activity by methanogens produces gas inside the biogas plant. A biogas plant usually consists of a concrete tank that is 10 to 15 feet deep, where a slurry of dung and other biowastes like plant and animal remains is placed. In fact, the biowastes and cattle dung in the slurry are first soaked in water and mixed in the mixing tank before they are allowed into the main tank, so that they provide the bacteria a suitable medium to grow. Methanobacterium and other anaerobic bacteria present in the slurry decompose it to release a mixture of methane, carbon dioxide, and hydrogen. This mixture of gases is also known as biogas. Moreover, the lid also collects the gas generated, which is allowed to flow through the outlet pipe, while the overflow tank collects the excess gas released. The biogas generated reaches nearby houses via an outlet pipe. Where it is used for cooking and lighting. The remaining exhausted slurry can be used as fertilizer. Did you know that the technology of biogas production was developed in India by the Indian Agricultural Research Institute, or IARI, and the Khadi and Village Industries Commission, or KVIC? India needs more such biogas plants to have an alternative energy source for LPG, since biogas is a low-cost fuel that burns without emitting smoke. As cattle dung is available in abundance in rural areas, many more biogas plants are being set up in our villages. However, even in cities like Pune, there are some commendable instances of lighting streetlights with biogas. Biogas can also be used to run electric engines such as pumps, as it causes less air pollution. Therefore, whether a village or city, establishing a biogas plant is an effective way to generate energy from wastes, and this has been made possible by microbial activity.